Yeah, so thanks a lot. Again, I'm Fabio. I'm a postdoc at Imperial College, but today I'm going to talk about what I've been doing during my PhD at the University of Leeds. And the title of my talk is Single-Cell Nanobiopsy Enables Multigenerational Longitudinal Transcriptomics of Cancer Cells. As I said, I'm a big fan of the Live-seq and what Oran and the other people did, and their work was very inspirational for me throughout my PhD. So, again, I don't want to repeat what Oran and Carmelo already said in the previous talk, but at the moment, single-cell RNA sequencing has a very static nature, so we can get information about the transcriptome of the cell in the present state, but then we need to lyse the cell, so we kill it, and we lose the possibility to follow and probe the transcriptome of the same cell over time. This can be particularly limiting in diseases such as glioblastoma, which is the brain cancer I've been studying. And glioblastoma is a very aggressive brain cancer because it has a tumor recurrence of 100% after treatment. And this is due to all these cellular subtypes in the primary tumor. So the tumor is highly heterogeneous. And when we apply standard treatment of chemotherapy and radiotherapy to the tumor, some subtypes, like the cyan one here at the top, they survive because they possess an inherent resistance, while some others, like the green one here, they somehow make a switch and they reprogram in order to resist to treatment. But if we want to understand and study this mechanism, we need to work at the single-cell level, and we need to be able to follow and probe the same cell here at the interface of treatment. So my study, the aim of my study was to develop a technique that allowed to take a nanobiopsy sample from the cell on day one here, from the primary tumor, do RNA sequencing, then treat the cell with chemotherapy and radiotherapy, and then 72 hours after the first biopsy, go back to the same cell or the progeny and take a longitudinal nanobiopsy and do RNA sequencing. But to do so, the technique has to be able to extract enough mRNA to allow the reverse transcription and amplification, to preserve cell viability, and to track the same cell over time. So I'm not a FluidFM user, so sorry about that, but I'm a scanning ion conductance microscope user. So scanning ion conductance microscopy is in a way similar to FluidFM, but rather than having a cantilever, we have a glass nanopipette, and this glass nanopipette is filled with an electrolyte solution. And there is an electrode inside this pipette. And the pipette is mounted on a Z piezo positioner and a DC motor. And the dish with the cells is put on a stage, and the reference electrode is put on the dish, and the stage is put on an XY piezo and XY motor for XY positioning. And everything is set up on an inverted microscope. So when the nanopipette is immersed in the medium, we have an ion current flowing between these two electrodes, and this current mainly depends on the pipette resistance, which is given by the size of the aperture of the pipette, and the access resistance, which is a measure of the occlusion of the pipette. So this access resistance is dependent on the distance from the sample. What does that mean is that our pipette is a proximity sensor. So when the pipette is far, it's in the bath solution, far from the surface, the ions are free to move in and out, and the current is high, so we are in this white region of the plot. But then when the pipette approaches the surface, the ion flow is hindered, so the current decreases, and we are in the green region of the plot. Now, if we select a set point and we stop the pipette at a specific ion current drop, and we scan the pipette in X and Y, we can measure the topography of our sample. So in this case, what I measured was the topography of hippocampal neurons. So you can see in high resolution the soma, all the dendrites, and you can also see the synaptic boutons. And if we do this on the cell line that I've been working as like a proof of concept, the glioblastoma cell line, these are the kind of maps that we can get. So we can see the cell. In this case, the cell was plated on a gridded dish, so we can see the details of the grid and the 3D topography of the cell. But the scope of my project was not imaging. I wanted to functionalize the probe in order to allow simultaneous injections and extractions from single cells. So the probe that I used is here depicted. It's a glass nanopipette which has 2 barrels, so it's a double barrel pipette. In one barrel there is an organic phase, it's an oil phase, and this barrel is used as an electrochemical syringe because by modulating the potential between this electrode and the reference electrode, we can electrowet the oil phase and we can cause a shift of the oil aqueous, so cytoplasmic phase, in order to cause an inflow of the cytoplasmic contents in the pipette. And the second barrel, I used the second barrel for 2 purposes. One was to record the ion current to be sent to the feedback control of the SICM, so this allows automatic positioning and operations. And the second was to inject a fluorophore inside the cell. The reason why we wanted to inject a fluorophore now was because to make sure that the biopsy was taken inside the cell. So we want to see the cell fluorescent before taking the biopsy, because we found that when you take the biopsy in the medium, this can lead to a lot of bias that is very hard then to remove from the analysis. And these are the probe, how they look like in the SCM. This is 150 nanometer approximately, the aperture of each barrel. So this is how the experiment looks like. So we plated the glioblastoma cell on a gridded dish in a non-equal ratio of wild type and GFP transfected, because we wanted to be able to identify a single GFP cell in a specific position, and then we wanted to take a biopsy and inject the cell with a fluorophore. So after the biopsy, one sample is collected and the cell turns yellow in consequence of the injection of the red fluorophore. Then we left the dish untreated as control, or we applied treatment, chemotherapy, radiotherapy, and 3 days later we went back to the same position on the grid and we took an equivalent number of nanobiopsies as the number of cells that we found in that location. So in this case, 4 cells are there, 4 biopsies are taken, and the 4 cells gets yellow after the procedure. So this is how the experiment looks like. So this is an example of a biopsy of a cell that was identified on day 1 and then was biopsied and injected and becomes yellow in consequence of the biopsy and the injection. Then the cell was left untreated and didn't divide over time. And then the cell was still alive on day 4, and we took a longitudinal nanobiopsy, and after, again, injection, the cell becomes yellow. And you can also see the biopsy mark here in red. Again, similar case of a cell which was biopsied and injected on day 1, and then was left untreated and divided. And then on day 4, we took 2 longitudinal nanobiopsies from the progeny. And again, an homologous case of a cell which was biopsied and injected on day 1, and then was treated with chemotherapy and radiotherapy, survived treatment, didn't divide, and then was biopsied and injected again. And then, again, final case, same thing, of a cell that was biopsied and treated, and then divided over the 72 hours, and then more than 1 longitudinal nanobiopsy was taken on day 4. Then, of course, this is only the sample acquisition. Then, as Oran already comprehensively explained, there is all the reverse transcription, amplification, library preparation, and sequencing. And to do this, we used SMART-seq2 because Live-seq was not already out at the time when we were improving this protocol. And we generated 2 libraries. One was a control library with whole cell lysate of treated and untreated sample, while the other was the library containing all the nanobiopsy samples. Well, from the sequencing metrics, we can clearly see that the nanobiopsy samples are affected by a lot of noise, so the data quality is not great. We can see it from, for example, the number of expressed genes, The nanobiopsies are here divided between day 1 longitudinal treated and longitudinal untreated, and then the whole cell are divided between treated and untreated. We have a lower number of expressed genes in the nanobiopsy. We have definitely a lower percentage of mRNA bases compared to the positive control. And also, we had to use low stringency criteria to do the filtering. So about 50%, 47% of the biopsy samples were filtered out compared to the 17% of the whole cell samples. But we continued and we tried to do dimensionality reduction, and we plotted our results on a UMAP coordinates. What we could see from the whole cell is a sort of separation between the treated samples at the top and the untreated, and an overlapping region here where both samples lie. This confirms what we found in the past, that we always get this kind of plots between treated and untreated samples. But what was interesting is that despite the higher level of noise, we were still kind of able to see a separation between the treated and untreated longitudinal samples. But I want again to highlight that the amount of noise was considerable, and that this didn't allow us to do the dataset integration as Oran showed in the Live-seq. So what we could only do was try to follow the cell on the UMAP plot in order to see how this moves across the plot. What we could do was to do a higher level analysis. So we went to the literature and we took the most established glioblastoma model for a transcriptomic model, and this model says that the two main subtypes of glioblastoma, so the proneural subtype and the mesenchymal subtype, they lie on the same axis of variation. So basically here PC1 is able to discriminate the proneural subtype from the mesenchymal subtype. So we took the gene sets and we did gene set variation analysis, because this was higher level analysis, and we took the paired untreated day 1 and longitudinal sample from the cell, and we could see that by assigning the proneural and mesenchymal score, the cells tended to switch subtype over time. So from day 0 to day 4, they underwent a subtype switch. But when we compared our treated samples, well, we couldn't see this. We've seen the opposite trend. So the cell tend to maintain the same scores, subtype scores, over time. So for the treated cell, it seems like treatment is kind of selecting for the cells with the most stable phenotype. Well, of course, this is still very preliminary data of a new technique. But I mean, I just want to highlight again the importance of this kind of work, and what Cytosurge and Oran did with the FluidFM, because the only way that we have to target this kind of diseases, this kind of biology, is to actually probe the transcriptome of the cell over time, and there is no other way at the moment to do this. So I hope that, I just want to thank my previous PhD supervisor, Dr. Paolo Actes and Lucy Stead from the University of Leeds. Georgette Tanner was the bioinformatician who helped me with the data analysis, and Chalmers, he helped me with the experimental design. These were my two groups, bioelectronics and glioma genomics. I want to thank Iain from the Harlem Institute, who helped us to improve the SMASIC2 in order to get to be tailored to our approach. And again, I want to thank, of course, Cytosurge for inviting me, and thanks a lot for the attention. Yeah.